Okay, so let's run through the key things that we need to know about key area 1, which is all on division and differentiation in human cells. So the first thing that we talk about are the differences between somatic and germline cells. So a somatic cell is any body cell that has nothing to do with reproduction, and they are always diploid. So they always, in humans, will have 46 chromosomes or 23 pairs of chromosomes. And because of that, they will always divide by mitosis to maintain that diploid chromosome complement. And in doing so, they will form and regenerate more of the same somatic cells. Okay? In contrast, germline cells are anything to do with gametes and gamete production. So they include two main types of cells. The gametes themselves, so in humans, the sperm and any ova, but also the stem cells that will divide to produce gametes. So the stem cells are going to be diploid. They have got 23 pairs of chromosomes. The gametes that they form will be haploid, and they will have 23 single chromosomes. Okay, so just to give you a little bit of an insight into what these germline cells are, when you get into Unit 2, you will start to look a little bit more in terms of gamete production. So you'll come across, within the testes, you'll come across these areas, these structures called seminiferous tubules, where sperm actually will develop and mature. And these things here quite often are called mother cells. They are the diploid stem cells that will give rise through division to the gametes. So these are diploid, these are germline cells because they give rise to the reproductive gametes. Okay, so as with any cells, they will want to reproduce themselves, make exact copies, and therefore they will divide by mitosis to produce copies of themselves. But they will also divide by another mechanism called meiosis. A meiosis involves two different divisions that will ultimately give rise to the actual haploid gametes, or sperm in this case. Okay, so we've got the diploid stem cells divided by mitosis to regenerate, but also the diploid stem cells divided by meiosis to produce haploid sperm cells. Okay, and the same kind of thing happens within the ovaries to produce ova. Okay, right, so just to summarize, somatic cells in humans are going to carry 46 chromosomes. They will divide by mitosis to produce more somatic cells, each with that diploid chromosome complement. Okay, however, in germline cells, the germline stem cells will start with 46 chromosomes. They can divide by mitosis to regenerate themselves to maintain the diploid chromosome complement. However, they can also divide by meiosis to produce these haploid gametes. Okay, so just to briefly, because this comes up in the specification notes for the course, meiosis, the process by which we go from the germline stem cell to the haploid gametes, it involves two divisions. So here would be our germline stem cell. The chromosomes are replicated, so you end up with these homologous chromosomes, and the first division is going to separate the homologous chromosome pairs. Okay, the second division is then going to pull those sister chromatids apart. So you've got two separate divisions. Okay, the next part of this key area looks at cellular differentiation, and this is how we start off with our gametes. They fuse during fertilization, or the nuclei do, to form a fertilized diploid egg. That will then undergo numerous mitotic divisions to produce other copies of that cell. However, we know that in a complex multicellular organism, we've got a variety of specialized cells, and that happens through the process of differentiation. Okay, when this happens, this is due to different genes being expressed to produce different protein complements in that particular cell. And by having different proteins, we end up with different specialized functions. In all embryonic stem cells, different genes can be switched on and off so that expression can happen or not. Okay, an example would be here. So we've got a bone marrow stem cell. So this can give rise to any cell to do with the blood. It can divide by mitosis and undergo selective gene expression and give rise to a whole host of different blood cells. Okay, so for example, in phagocytes, and the cells that will eventually become phagocytes, that bone marrow stem cell will have genes switched on to produce digestive enzymes. Okay, cells are found in the very early embryo, and they still have that potential to differentiate into any cell type, and because of that, they are known as pluripotent. Okay, so here, embryonic stem cell, we can remove one of those, culture it, and providing different signals to encourage selective expression of different genes, we can encourage the differentiation into a host. In contrast, tissue stem cells have a little bit more of a limited repertoire of cells that they could differentiate and specialize to become. They tend to be involved in growth, repair, and renewal of the cells found in that particular tissue. Okay, so for example, a bone marrow stem cell would be involved in regenerating and repairing new blood cells. Because they cannot become anything, so for example, a bone marrow stem cell could not give rise to a new nerve cell. It can only give rise to new blood cells. They are known as multipotent. So they can become multiple cells types, but not all. Okay, so multipotent, multiple potential to become any cell within that tissue. Pluripotent has potential to become all specialized cells, but we obtain it from the embryo. Now we can use them for different things. Now this is important that we differentiate between therapeutic and research uses. So in terms of therapeutic use for a stem cell, we want to be quite specific. So we want to move up from the NAT5 level and not just say, oh, it could be used to help cure diseases. We want to be specific. So in terms of a therapeutic use, it could be involved in the repair of damaged or diseased organs, for example, in corneal repair, or for the regeneration of damaged skin. So you would want to be specific in how it's involved in helping a particular, want to explore a little bit more around that idea of helping to cure a disease, and we want to have a bit more of a specific example. In terms of research use, there are numerous things that researchers might use stem cells for. We could use them to test drugs. We could use them to understand disease development a bit more, or learn how to come up with new treatments for disease. Okay, so we can use them as model cells to discover how disease develop, or can be used for drug testing. Okay, we can also use stem cells in research to understand more cellular processes, such as how do the cells grow, differentiate, and how does gene regulation actually take place. So these would be classed as our research uses, not our therapeutic, and SQ may need you to be able to differentiate between those two types of uses. And finally, we move on to the brief section that talks about cancer development. Now this you don't need to know, but as our cells go through a kind of growth cycle, they'll go through a very complex series of events. So they will divide. They will go through a period of growth. When they reach a certain level of growth and there's enough cell structures and proteins there, the DNA will then replicate. It will then go through another period of growth. Everything's checked and signed off and double checked again, and then and only then, where everything's checked, does mitosis take place. And you will see there are a series of checkpoints. So the process of mitosis and cell division and cell growth is a tightly regulated process, and it needs to be. And when that goes wrong, sometimes things like cancer can develop. So cancer cells will be those that divide excessively because they are not responding to regulatory signals. So each of these points here, we've got these regulation checkpoints that are controlling the cell passage from G1 to S to G2 to mitosis. So each of these wee sections of this cycle, it's controlled how easily it goes past. And when there's a regulatory signal given, the cell's allowed to move on. Cancer cells don't respond to that, so they divide excessively and it results in a mass of abnormal cells, and we call that a tumour. So here would be our mass of abnormal cells. It starts to form this big tumour, and eventually there might be some kind of visible lump underneath with quite an extensive blood supply. So our normal cell will divide. If there's something wrong, it will normally undergo a programmed cell death process. However, in cancer cells doesn't respond to regulatory signals, and it then doesn't undergo a death process. It continues to divide and form a tumour. The other thing that can happen is when that tumour is building up, so say we've got an original tumour here in the large intestine in the bowel, what can happen is that cells have this ability usually to attach and adhere to each other. But in a tumour, in an actual tumour, that sometimes does no longer happen. And one of those cells, one of those abnormal cells, may fail to attach to the rest of the mass and then spread through the circulatory system and eventually reach a secondary location. So it travels through the blood system and, for example, could attach and form a secondary tumour in this example in the brain. Okay, so it travels in the bloodstream after failure to attach to the original mass.